Hello everyone, I am Enrico Luchinat from the University of Bologna and the Magnetic Resonance Center in Florence. In this third part, I will show how to record and process the in-cell NMR spectra. First of all, we have a very limited time to analyze these samples, so virtually any liquid state NMR experiment would be okay to analyze your cell samples. However, you have a quite intrinsic low sensitivity anyways, because NMR is intrinsically insensitive, and then you are limited by the cell viability. And these cells last only a few hours at 37 degrees at such high density in the NMR tube. Then they will start to die. So we need as fast and as sensitive experiments. And these are most of the time the fast type of experiments, like the SOFAST HMQC on the best type 2D and 3D triple resonance experiments. Then there are other methods that I will not talk about to shorten the acquisition time. So this is how we do the setup for a few pulse programs that we use routinely to analyze cell samples. Now bear in mind that these steps, the initial setup of these NMR experiments, should be performed at any time before you prepare the actual in-cell NMR sample. That is because if you have cells ready in the tube and then you begin only then to prepare these NMR experiments, it will take some time, and that will decrease the viability of your cell sample. So you want to be quick when you have your cells ready for acquisition. So I would suggest to prepare this data set before you actually do the in-cell NMR sample. Now, so you first prepare an empty data set at the NMR spectrometer. We have top spin there. Then you set the parameters for the 1D NMR experiments. I always acquire short 1D for all my samples. That is because this is useful for comparison and to see how many cells I actually have in the pellet. That can be slightly different, and then it's a very useful comparison among samples. So to set up this 1D proton monodimensional NMR spectrum, I center the carrier. You center the proton carrier to the water signal at 4.7 ppm. Then this is the pulse program I use, the ZGESGP. So I set the spectral width to 20 ppm, but it can be 24, 30, and then a square or a sinus shaped pulse of 1 millisecond long for water suppression. Then I set an interscan delay of 1 second. It could be longer than that if you need. And then I normally acquire this with 128 scans that last roughly 5 minutes. So this comes before any other experiment that I use. Now sometimes I want to see some proton signals in the 1D spectrum that actually fall outside of the crowded spectrum of the cells, which means that I can observe my protein even on unlabeled samples, just by looking at the 1D proton spectrum. Now for this, I often use the P3919 pulse program, which has a water gate with a binomial water suppression scheme that has a particular excitation shape, which has a maximum at a variable region in the spectrum. So you set the spectral width to larger than 25 ppm in my case, and then you adjust the delay for the binomial water suppression. So that is explained in the pulse program, so that the maximum excitation is centered at the chemical shift of the signals that you want to observe. And then again you set the interscan delay of 1 second or more, and then in this case, as these signals are quite small, I usually do more scans, like 512 scans, and this lasts around 30 minutes. Now we come to the 2D 1H15N SOFAST HMQC. So this is a proton-nitrogen correlation spectrum, which is the equivalent, the fast equivalent of the HSQC or HMQC that is normally used in vitro to observe a fingerprint of the protein backbone. So the SOFAST, which stands for Band Selective Optimized Flip Angle Short Transient, it's a scheme which exploits selective proton pulses to only excite the amide protons. So these are band selective pulses. You see these pulse, this 90-degree pulse for excitation, and then you have the 180-degree pulse for refocusing. They are both shaped and they are band selective. This gives you the advantage that you are only perturbing the signals that you want to observe, and you don't touch the water signal, the water protons, which are many in a solution, in a water solution, nor the aliphatic protons. In this way, you cause a much faster longitudinal relaxation, and that allows you to decrease the interscan delay. That is how long you have to wait between one scan of the same experiment and the next scan. The magnetization that you want to observe will recover faster, and that allows you to just repeat quicker the same pulse program. Then there is another optimization that makes use of the nth angle alpha, which is normally 90 degrees. In this case is optimized. It's typical 120 degrees to maximize sensitivity at a higher repetition rate. So how we set up this? So we, well, usually start with the load, with the default parameter set for the SOFAST HMQC. Now in modern Bruker spectrometers, actually everything is mostly already set up. There are a few parameters that is your choice. So like the carrier frequency, we set it at the water signal at 4.7 ppm, and the nitrogen frequency, we normally set it at 118 ppm of nitrogen. Then I set a spectral width of 15 ppm for proton and 50 ppm for nitrogen. That can be narrower than that if you already know where the signals will fall in the spectrum. If you don't know that, then it's better to stick to a very large window on the nitrogen, and 2000 points and 128 increments in the indirect dimension. Then, this is very useful, on the newest top spin version, you have pulse programs that can calculate automatically the length and power level of the shaped, the band selective pulses. So it's useful to use the option decalc_sp so that it will calculate automatically these values based on just two parameters: the center of the band selective proton pulses that I usually set at 8.5 ppm right into the amide proton region, and the width of the excitation window. This is important to set properly so that you avoid to excite the water resonance. If this pulse is too wide, then you will start to excite the water, and you will then cancel the positive effects of using a band selective pulse for this type of sequence. Then I set an interscan delay of 300 milliseconds, which is much shorter than the usual 1 second delay. Then, now we have set up all the experiments. We set the desired NMR temperature, the temperature of the NMR spectrometer, which in my case is normally 37 degrees. And now for the real acquisition of the NMR experiments. At this temperature, in our experience, the cells remain viable for around 2 hours. You could make it a bit longer and acquire for 3 hours, but I would recommend stay within the 2 hours time limitation. Then you should always check the viability after the NMR experiment. So you have set up the experiments already and you have the samples ready. So you load the dataset with the desired NMR experiments. You insert the in-cell NMR sample into the magnet. Then you check the matching and tuning of the probe head, and then you shim the magnet. Now, you may read sometimes that you need to either manually shim the tube, so make the magnetic field homogeneous inside the NMR sample, or actually you have to first insert another tube and then shim that tube and then replace it with your sample. In my experience, it's really not needed. We can just use the automated shimming routine, top shim in the new version, and that works well directly on the inside NMR sample. So most of the time, even 3mm Shigami can be shimmed very nicely with top shim. So after shimming, then you calibrate the 90-degree hard pulse, and then that can be done manually or automatically. And then you adjust the power levels of the proton shape pulses accordingly, and that is usually automatic. Then you record the NMR spectra, and again, depending on how many scans you want to do, the total time of acquisition will change. In all these spectra, you will expect to have very broad lines, broader than 15 Hz, which is usually a lot for a high-resolution NMR experiment. However, that is expected and that is normal, and that's due to the intrinsic inhomogeneity of the cell sample. So it has nothing to do with the poor shimming of the magnetic field. It's because of the cell sample itself. Then during acquisition you can process, and then just to look how the spectra are coming, and then when they are finished you can do the actual processing, the proper processing. So to process the proton NMR spectra, I usually use an exponential decay function to multiply the FID, and the line broadening parameter can be set at your desired value. I normally use between 5 and 20 Hz. That's because I already have intrinsically broad line in the proton spectrum, so I can use an even broader function. Then zero field and Fourier transform like you would normally do to process NMR spectra. And then you can correct the phase along the proton dimension. And you will get from the FID to the actual cell spectrum, which is, if you can see this is actually in cell NMR spectrum. Then you process the 2D NMR spectra by, again, multiplication with a window function along both dimensions. My preferred one is the SSB, that is a square sine Bell function, with SSB value equals to 2, which is the 90-degree shift of the sine, which becomes a cosine Bell, basically. Then zero field and Fourier transform in both dimensions. And then you, again, correct the phase along the proton and nitrogen dimension as you would do with a normal NMR spectrum. And this is what you get. This is the spectrum of superoxide dismutase 1 with only zinc bound to the zinc binding site, by the way. And you can see that on top of the nicely resolved cross peaks arising from the protein itself, you also have a broad signal here, which is actually composed of many smaller signals. So these are thousands of signals, and this is the cellular background. So what's that? That arises because protein expression in hex cells lasts for 2 days, and in 2 days the N15 labeled amino acids will also be incorporated into other cellular components, because cells are still metabolically active and they make proteins and they divide and then they grow. So they will get eventually N15 labeled, and that gives rise to this background. Now, so on top of that, you also have some very strong signals like these and these that arise from very abundant small molecules, metabolites. In this case, glutathione. Now, to get rid of that, there is actually a very useful procedure. That is, perform an N15 background subtraction. Now, if you can almost eliminate this background almost entirely by subtracting 2 different NMR spectrum. One is acquired on the in-cell NMR sample containing the protein of interest. So this spectrum here, in this case, where the cells have overexpressed superoxide dismutase 1. The other spectrum is acquired on a different sample, which is a control in-cell NMR sample, where the cells were transfected with empty DNA vector, and then they are kept into the labeled medium for the same time as the sample where you express the protein. So if you subtract this will be just the N15 background signal from empty cells. So if you subtract this to this, you get this, which resembles an in vitro NMR spectrum, basically. So you got rid of most of the cellular background signals, and what remains is a high quality HSQC type correlation spectrum, in-cell spectrum. Now these are a few examples of what kind of spectra you would expect to obtain from different proteins. Now keep in mind that these are all unfolded proteins. However, some of these, like CCS or Maya40 here, they contain some loops or tails which are not structured, and then you can tell from the intensity of these signals and from the fact that they are all collapsed around 8.3 ppm in the proton dimension. That is because they arise from unfolded regions of the proteins. But also for folded proteins like HAH1 or SOD1 or a mutant of profilin 1, COP17, we get nice, high quality, and also quite high resolution NMR spectra. Now CCS is an interesting case because actually CCS is a large folded protein. However, the only signals that you see are these few signals arising from unfolded loops. That is because CCS is one of those proteins that interact strongly with other cellular components. And that's a perfect example to show what happens when your protein is not tumbling anymore like a real solute, but it's actually sticking to something into the cells. All the signals arising from the folded protein disappear. Whereas in the opposite case, you have SOD1, which is smaller but not that smaller than CCS, which does not interact with the other components, and you can see a beautiful spectrum. Now, those were acquired on uniformly labeled samples with N15. So all the amino acids contain N15. You would see one peak for each residue of your protein. Alternatively, you can use different labeling strategies to prepare cell samples. And for example, we have performed amino acid type selective labeling, such as N15 cysteine labeling, or C13 methionine, where only the terminus carbon is labeled. Now, these allow you to obtain much cleaner spectra where you don't even need to subtract the cellular background because only that type of amino acid will be labeled. So, for example, cysteines of SOD are 4, of which you can only see 3, however, which are these 3. And here I did not subtract any background, so you also see this big signal, which is glutathione, which contains a cysteine. And this is another example in which you clearly see 4 signals arising from the protein methionines, and then a 5th signal, which is free methionine. In some other cases, you can even avoid using isotopes, because sometimes your protein will have signals that fall into a region of the proton spectrum which is completely free from other signals from the cellular components. That would be flat in a control sample, which means that you can just acquire a proton spectrum with a binomial water suppression scheme, for example, and observe just 1D proton signals of your protein inside the cells. Then after the NMR experiment, it's useful to perform a trypan blue exclusion stain, which is a very quick way to assess how many cells are damaged, and so they are dead. So permeable cells, damaged cells are permeable to the stain, and so they will stain in blue, whereas healthy cells will not be permeable and will not be stained. So then you can count the cells under a microscope, and then you count the blue cells and the total cells, and you get a percentage of the cell death. You should then perform further analysis via NMR. One of these is the analysis of the supernatant, which is the NMR buffer that you used. So to do this, you need to take back the cell sample from the tube. To do so, you take again the glass pipette, and then you slowly resuspend the pellet at the bottom of the tube in the medium itself that was still in the tube. Then you collect everything, you put them in an Avenger tube, and you centrifuge them at 800 G for 5 minutes. At this time, you can collect the supernatant which should be clear solution. Then you put it back in an NMR tube, and then you record the same NMR experiments that you acquired on the cells. You should not observe the proteins, the signals from the protein of interest. If you do observe these signals, and if their intensity is comparable to the intensity of the same signals in the in-cell NMR, it means that the protein leaked out of the cells. This is bad because it means that what you thought you would observe, that is the intracellular protein, actually was protein outside of the cells, even in the in-cell sample. So that is a problem. It means that the cells are either dying or the protein is somehow leaking from the cells. And so it means it's very important to do this control experiment, because it should be used to validate that the actual in-cell NMR experiment. And then you can perform a cell lysate and then observe the cell lysate, analyze the cell lysate by NMR. To do the lysate, you resuspend the pellet in 150 microliter of PBS, for example. A lysis buffer would be okay, any other lysis buffer. Then you lyse the cells by freeze-thaw cycles. That is, we use liquid nitrogen to freeze the pellet, and then water. Then we just alternate between nitrogen and water, so that each time we snap freeze, we break the cells. So around 8 to 10 cycles like this, you will have destroyed all the cells. Then you spin down this the lysate at maximum speed, in our case 16,000 G for 1 hour at 4 degrees. And then you collect the supernatant, which should be mostly consisting of the cytosol of the cells. Then you can put that in the NMR tube and analyze. And now this is a slide to explain you the workflow that we normally apply with when we perform in-cell NMR on a protein that we never observed before. So first, we should check the expression level by SDS page on some other control samples. Then suppose that we have good expression. We perform the in-cell NMR experiment. Do we detect the protein? If we do detect the protein, like in the previous cases I showed, that's nice. It means that the protein is behaving as in solution. That is, it's free to tumble, it's not sticking to anything, and you can see beautiful spectrum. If you don't detect the protein, then you should first perform the NMR on the lysate. So you extract the protein, take it out of the cells, and then do you see the protein? If you see the protein, it means that the protein was in the cells, it didn't leak out before you prepared the lysate, but it's still interacting. It means that inside the intact cells, it was interacting with other cellular components, and that would cause a slowdown in the tumbling rate and then a broadening beyond detection of the signals. If you don't detect the protein even in the cell lysate, then you should check by SDS page or by Western blot both the supernatant of the cell lysis and the pellet of the cell lysis, because it could be that the protein actually was not remaining in the soluble fraction of the lysate. And maybe that can happen if the protein is aggregating inside the cell, or if it's sticking to some other components like the membranes or the chromatin, for example. So if you detect the protein in the soluble fraction here, then it means that the protein was interacting so strongly with some other large component which was also soluble that you could still not see the protein. You still would not see the protein even in the NMR lysate sample. Then if you don't detect the protein in the soluble fraction of the lysate, it means that either is not soluble, so that it remains in the pellet, or that the expression level, in this case, if you don't detect the protein in both fractions, it means that the expression level was too low for these particular experiments. Maybe something went wrong in the transfection. Sometimes that can happen, and then you should repeat the experiment. So I hope that was useful and understandable, and these are just a few references that can be useful to read and to if you are interested to know more about what we did with this approach, direct protein expression approach in human cells at CERN. So these are a few works where we observed events like protein folding, maturation, and aggregation of pathogenic mutants. Then we could also target the protein of interest to organelles. We did that for mitochondria, when we could purify the mitochondria and observe the protein directly inside the mitochondria. We can use this approach to investigate the redox state of proteins, for example, the formation of intramolecular disulfide bonds as a function of the environment or as a function of co-expressed cellular partners. And then recently we showed that we can apply this approach to observe intracellular ligand binding to the protein of interest. And that means that in cell NMR on human cells can be applied to perform protein-observed ligand screening, potentially useful to investigate properties which are correlated to the potency of potential drugs. So I hope that was interesting for you, and thank you.